Hello, everyone. This is Bergmann. Let me start to share my screen to start my presentation. I want to welcome everyone, and I want to thank also the organizers for inviting me to give this presentation. I'm particularly excited to share some of our work with new postdocs and graduate students and users of these amazing X-ray lasers. The title of my talk is called Stopping Time, the Emerging Science of Powerful X-ray Lasers. Before I start my talk, I want to make a little pitch. This is my group in Wisconsin, and you can see at the bottom, you can see our website. We are interested in different areas of research. Most recently, we have obtained funding to investigate nonlinear phenomena and to develop new X-ray laser methods. And I want to pitch the fact that we have a postdoc position joined with SLAC and Stanford, which is open. And if anyone is interested or if anyone knows someone who might be interested, please contact me. We are really excited about this. This is a three-year funding for that position. The title of my talk, Stopping Time, was actually very much inspired by a book from 1934 called Stopping Time. Henry Chapman, my colleague, at one point recommended that to me. And there is a specific picture in this book. This is a book where they used flash photography to really stop time and show actions which usually are way too fast to see. And in this particular example, it's an archer who releases an arrow. And what happened, he accidentally angled the bow in the wrong direction. And then when he released it, the string carved into his arm. And you can see that here. You can see how this string carves into the flesh, and this is very painful. And the interesting thing about this picture is that when the picture was taken, in the instant of this photo, the archer didn't really know yet, because it takes about maybe 100 milliseconds or so for the signal to reach the brain. And in a sense, we have probed this person's action before the damage has set in. And you will see that this is very similar to the concept of how X-ray lasers are used actually to probe, to stop time and probe materials before these powerful X-rays can actually damage the material such that you lose the information. And also, this happens actually quite often. And here you see the Korean superstar from the Tokyo Olympics, An San, 20-year-old, and she won 3 gold medals for the first time. And like others, she uses this protection. You can see that I spotted that on one of the coverages, and she uses this protection to prevent this painful thing to happen. So here you see an exposure time of a microsecond. This is a bullet going through an apple, and it's really amazing the detail you can see. Normally, you would only see that this apple has been obliterated into 1,000 pieces and nothing is left from it. But originally, first, before all that happens, it actually rips a hole through the apple before the rest of the damage actually happens. Now, if you want to go much faster than that, so this is 10 to the minus 6 seconds. You might, for example, be interested in a water molecule. So we know water molecules they look like this, but in reality, they're never at rest. They vibrate, and they have these all these known vibrations, different modes of vibrations. And imagine you wanted to stop. The motion of such a vibration. You would need to have 8 orders of magnitude faster than what you just saw with the apple. You would need 10 to the minus 14 seconds, or as we call it, 10 femtoseconds. And that is exactly the length scale of X-ray lasers. And it was first 10 years before X-ray lasers really started. It was proposed by Haidu and Neutze and others that you should be able to image a molecule. So this is a molecule with an intensive X-ray laser pulse. And the time it takes for this molecule to blow up, so the time it takes for the damage, should be such that if you have 10 or so femtosecond short pulses, you should be able to outrun this. This concept, probe before destroyed, has also been shown by many experiments now, is really at the heart of X-ray laser research. Many of the samples are such, in particular chemical and biological samples, that they're replaced for each shot of the X-ray laser. It was Claudio Pellegrini from UCLA and SLAC who originally proposed to turn this SLAC LINAC into an X-ray laser. And here he is after his award, the Fermi Award, very prestigious award from the Department of Energy. And here he discusses his ideas of what he proposed with President Obama at a visit. So let me just show you briefly a video how the X-ray laser at SLAC actually works. This is the famous LINAC, Klystron Gallery, and we are at kilometer 2, so 1 kilometer before the end. This is underground, 25 feet underground. You have a laser hitting a copper cathode, knocking out about 10 billion, 1 billion to 10 billion electrons and moving them into the main accelerator. They get twice compressed with a bunch compressor to keep them really close and tight together. And then they get accelerated to full speed. It can be as high as 15 GeV, so it's somewhat between 3 and 15 GeV. And then they go through the so-called transport hall into the undulator hall. And in the undulator hall, these electrons are forced by a series of magnets onto a slalom course. And here they start to first emit synchrotron radiation. And then this radiation stimulates the emission of other radiation. And in a process called SASE, it orders actually the electrons to emit coherently. And this SASE process, standing for self-amplified spontaneous emission, creates these very, very bright pulses. And they travel together with the electrons, and then basically the electrons are guided off into the ground. We don't need them anymore. And the X-ray pulses enter the various experimental stations. This is the original one, which had 6. Now we have about 8 at SLAC, even also on the second floor. And this is where these experiments, these amazing experiments with these X-ray pulses are carried out. And this is a cartoon of one of those instruments, what you see here. There are now, in addition to SLAC, there are 5 other hard X-ray lasers around the world. There's several more with a soft X-ray. I just show you the large hard X-ray lasers, the European XFEL outside of Hamburg, the Swiss FEL close to Zurich, the Shanghai FEL, this is relatively new, it just got lasing for the first time a little bit more than a year ago, and the SAKLA in Japan and the PAL FEL in Korea. You notice that each of them also has a synchrotron close by. You can see here, this is the Swiss FEL, the Swiss light source, you have the Shanghai synchrotron, you have the Pohang synchrotron right here, you have the big Spring 8 here, and this one you cannot see because it's in the distance. The Petra 3 ring in Hamburg. LCLS has been upgraded now to a so-called LCLS 2, which has 2 linear accelerators, including the new superconducting accelerator. And the really importance about the superconducting accelerator is the fact that you can actually produce high repetition rates because there's no resistance, so you can produce something like up to 1 megahertz. Normal so-called warm linear accelerators, they only produce repetition rates up to 120 hertz, 120 counts per second. The Hamburg one is also superconducting, so that one can also go to high repetition rates. And this is a view of it. You can see the helium the center where the big helium cryostat is, and then you have several pictures of this is the linear accelerator cells, and then these are the two undulators side by side, as we have two undulators, the soft X-ray and the hard X-ray undulator. And here's a cartoon of how these experiments are done. You drop your sample into the X-ray beam, and then you create either diffraction or scattering or spectroscopy signal, and you record this shot by shot. And in this particular case, it's a scattering signal, and then you use that information to learn about your structure or about your chemical signature of your sample. I will share with you a few short experiments. This is a beautiful experiment done by my colleague Anders Nilsson, and he measured, basically he initiated the CO oxidation, carbon monoxide oxidation, similar to what happens in a catalyst, and he would use spectroscopy to stop the time to make a movie of this amazing reaction. Here you see the cartoon of it. The heat of his laser pulse starts to bring in motion the possibility, and then actually the CO is kind of hovering over it, and then finally finds its partner and comes off as a CO2. This is what happens in a catalytic center, and Anders was able to basically stop the time of this thing, which the total length scale is about a picosecond. So that's really, really short. Another example, which I really like, is the vitamin D synthesis. This was done by my, this is a very important reaction, which actually happens in the human skin. We make vitamin D ourselves. It's, in that sense, vitamin D is not really a vitamin, it's a hormone. We call it a vitamin. And what happens is that the initial reaction of vitamin D synthesis is when UV light, in the case of the sun, breaks this cyclohexadiene ring, which is a carbon ring. And once that ring opens, vitamin D can be synthesized. And so my colleagues led by Mike Minetti at SLAC, and also Peter Weber at Brown University, what they did is they used these cyclohexadiene molecules. They dropped them into the X-ray laser beam, similar to what I just showed you with this molecule. And before they shot them and measured the scattering signal, they excited them with UV light and see how these rings are opening up. And from the scattering signal, they then use that information to look at how these rings are opening up. And the three most likely ways how this super fast ring opening happens is shown in this cartoon, which is it's based on the data and on many, many simulations, on many traces of simulations. And they find that these are the most likely ways. So in one case, actually, the ring even closes again and then opens. They haven't stopped the time. These are really short movies. These are only maybe a few hundred femtosecond, the whole movie. So very, very fast. Another example is what we have been doing in photosynthesis. And this is work which I've been involved in myself for many years, large collaboration led by Lawrence Berkeley Lab, and there are many other collaborators. And we basically are interested in what happens in photosynthesis, how when we use sunlight to split water to create the molecular oxygen, all the molecular oxygen in the atmosphere. And the experiments, they evolved over time. We've originally injected these small crystals of photosystem 2 protein in a jet, and more recently have developed a system where they are injected in a droplet injector. They are then exposed with a laser which simulates the sunlight, giving different pulses, because this reaction the full reaction requires more than one pulse. It's up to 4 pulses. And we then, as a function of time, we then stop and measure the diffraction to get the overall structure, and also the spectroscopy to get the manganese chemical structure during this really important reaction. So this is a cartoon of how how this is done. So to the left, you see the old system, and to the new, this new injector. Crystals are in these little droplets, and we fire them with a laser, and then expose them to either 1, 2, or 3 flashes, and then look actually with an X-ray pulse shortly after the last flash. And we can change the time delay, and we can record in this big detector, we can record the diffraction. So we record many, many diffraction spots, and then get the 3-dimensional structure. And in this one to the left here is a cartoon of our spectrometer. There we can measure the manganese oxidation state simultaneously as we are getting these structures. And this work has been ongoing for many years now. We have published a series of many papers. Every time we start to improve the resolution and the amount of information. And I'll just show you one of the steps. We now believe that here's a series of shots. These are the oxygens. These are the manganese. This is part of this manganese cluster. And we have seen now that there is a so-called OX, an additional oxygen showing up. And we believe that this is related with the formation of oxygen. Whether it's these two or in a different way, or whether it replaces it, that is still an open question. But we have now stopped the time of photosynthetic water splitting, and we can capture this. This is not a very fast reaction, but we do need an instant probe before this destroy mechanism of an X-ray laser to actually capture this. If we wanted to measure that with a synchrotron, the amount it takes to accumulate the data would be too long, and we would smear out the effect, and we would also be prone to a lot of damage. So the X-ray laser in this case has been used to really use that principle shown first by this Archer probe before destroy. So that allows you now to do these measurements also at room temperature and not only at cryogenic temperature. It's really one of the wonderful new capabilities of X-ray lasers. You can measure samples at room temperature because the pulses are so short that there's no time for normal radiation damage to occur. So here is a picture of, this is a typical group size at the beamline at the end of this experiment. This was before COVID. Now in between it changed a little bit. Now we are coming almost back to full strength. The authors on the paper are even more than this. We have typically 30, 40 authors. But for those of you who are interested in LCLS and X-ray laser research, that's kind of the group size. And you see many of my colleagues, and of course, the leadership from Lawrence Berkeley Lab, Vittel in the background, Junko and Jan. And you can hear Jan and a lot of young people, and you can see that it's actually a very, very exciting place. X-ray lasers are really exciting places to carry out research. And these experiments are complex, large experiments, and they require a large team and long preparation. And but it is really an enormous amount of fun. I want to guide a little bit for those who are interested in some further reading. This is a review which we recently published, and you can actually upload it. If you go to my homepage at Wisconsin, you can upload the PDF file of this review. And this is specifically written, it's a new journal in Nature called Nature Reviews Physics. And it is specifically written on a collegial level. So if you're a new researcher in this field, this is completely understandable. And I'm very happy to have been part of this great team. This is Vidya Chandra from Lawrence Berkeley, Junko Yano, and also Jan, and you have seen them already before. And this is Bob Schoenlein, the LCLS Deputy Director, and this is Philip Bernard, my old friend and colleague. He's a professor at Uppsala, and he has done some groundbreaking work with soft X-ray, Rick spectroscopy at FES, and a lot of other work. And we have been collaborating for many years. And I want to finish with a quote. And this was a quote by the director of the Riken Spring 8 Center, which operates the FEL in Japan, the Sakla FEL, and also the large synchrotron, the Spring 8 synchrotron. And the director is Tetsuya Ishikawa. And we were able to actually get his quote in a book which we published recently, a few years ago, on X-ray free electrons laser. In fact, Vittel and Junko and myself, we are the editors of this book with the contribution of many of the pioneers, really wonderful contributions of the pioneers. Again, this is something for the reader who wants to get into this field is very much recommended. And the quote goes as follows: "The basis of science is observation. The most fundamental way of observation is by light. X-rays are a form of light that can probe atoms and molecules. X-ray lasers can probe atoms and molecules on the fundamental timescales, femtoseconds. In other words, they can stop time. X-ray lasers are therefore one of the fundamentally most powerful tools for science." And with that quote and with that exciting quote, I would like to thank you. And I'm very happy to take some questions. And I'm also happy to discuss with you any further collaborations or any other questions you might have about X-ray free electrons. Thank you very much.